I am waiting here for the initiation of class, and the students and viewers kindly give me message for the confirmation. One and two students give me message. I am waiting. The students connected. The students who are connected kindly give me message. And confirm that my sound is audible to you. अगर आप लोग तक मेरी आवाज़ जा रही है तो मैसेज कर दें. तो मैं now I am initiating my lecture on Ramachandran Plot. It is in your syllabus, that is in the syllabus of the MSc prescribed by the universities of Bihar. And of course, the topic is present in the csir-net examination, and it is the significant topic both for the MSc and net examinations. So I am going to explain fundamentals of the Ramachandran plot. In my last class, I explained that the proteins are present in primary structure, secondary structure, tertiary structure, and quaternary structure. Of course, we all know that the proteins are formed by the polymerization of amino acids. And in my last class, I also said that the peptide bond is formed between carboxylic group of amino acid associated with the alpha carbon and amino group of subsequent amino acid also associated with the alpha carbon. So, in the peptide bond, the carboxylic group and amino group are associated with the formation of peptide bond. And you all studied in your intermediate class that the organic molecules acquire a spatial arrangement. You all studied the methane, and in methane you observed that the methane represents tetrahedral nature. So, the one carbon is associated with 4 hydrogen, and the hydrogens are associated with a definite angle with the carbon to form the spatial arrangement. Likely, in the same way, if the peptide bond is formed between the carboxylic group and amino group, again the first amino acid, then the second amino acid, then the third amino acid, in this way, by the polymerization, amino acid form the polymeric form, and then the polymeric form is again formed the non-peptidic bonds and represent primary, secondary, tertiary, and quaternary structures. But the molecules associated with the peptide bond in amino acids represent a spatial arrangement, and that particular spatial arrangements of molecules are responsible for the spatial arrangement of the molecules of protein. So, the bond length and bond angle associated with the alpha carbon are necessary to understand properly that in which form, in which nature they are arranged in a space, and that a spatial arrangement of the associated nitrogen, carbon, oxygen, and hydrogens at the peptide bond is represented by Ramachandran plot. And Ramachandran plot was at first presented in 1963 by Ramachandran, and Ramachandran plot is also referred as Ramachandran diagram or phi psi plot, because we commonly use the bond by the Greek letters, and the letters used for the explanation of nature of bond, the symbols which are used for the explanation of bonds are phi, psi, omega, and omicron. These four letters are used for the representation of a spatial arrangement of molecules in bidirectional mm, form, but they represent the spatial arrangement, and then particular representation is referred as Ramachandran plot. The Ramachandran plot was explained by G.N. Ramachandran, C. Ramakrishnan, and V. Shashi Sekharan. These three are scientists. I am again repeating: G.N. Ramachandran, C. Ramakrishnan, and V. Shashi Sekharan. And these three workers explained the Ramachandran plot, or explained the method for the representing of a spatial molecular arrangement in the proteins at polypeptide bond is referred as Ramachandran plot or Ramachandran diagram or psi-phi plot. Psi is represented, just you form a O and cross it, that is the phi. Psi is represented by a trident, and omega is represented by a small w like a structure, and omicron is represented by a small o. And these four are the symbols for the phi, psi, omega, and omicron. The Ramachandran plot provides a simple two-dimensional graphical representation of all possible protein structures in terms of torsion angle. What is torsion angle? Just I am going to explain in this class. The torsion angle. What is the torsion? Torsion angle. And the torsion angle is represented the all possible structural diagrammatic representation. Although The plot is developed using theoretical and mathematical or statistical method. The plot is developed by the use of theoretical and mathematical or statistical method. It is highly significant. The Ramachandran plot is highly significant for the study of a spatial arrangement of molecules in polypeptide chain. Because in last class I told you that the spatial arrangements or spatial organization of the molecules present in the primary polypeptide chain is responsible for the differentiation of proteins into primary protein, secondary protein, tertiary protein, and quaternary protein. So here the torsion angle or Ramachandran plot is specifically determines the nature of a spatial arrangement of molecules in any specific protein configuration. So it is most important for the configurational study and configurational analysis of any protein. Here, the if you observe the peptide bond, it represents planar nature. It represents the planar nature. For example, one in last class also I explained in my last class also that in one amino acid the carbon alpha carbon is associated with amino group, carboxylic group, and hydrogen and one alkyl, except in glycine and proline, because in glycine R is replaced by H, and in proline the imino form is present in place of amino form. So the configuration only two amino acids represent some variation in this fundamental configuration of the amino acids. So the one amino acid, when associated with the subsequent amino acids, then carboxylic group H of carboxylic group is associated with the OH of carboxylic group is associated with the H of nitrogen, that is amino group, and removed in the form of H2O, and now it forms the peptide bond. The peptide bond is represent the planar structure. Here the peptide bond represents the planar structure. If we observe the planar structure of the amino acid, thermodynamically the formation of peptide bond is endergonic process. It requires the energy, endergonic process, and the delta G0 value is plus 21 kilojoule per mole per peptide bond. Here, in my previous class, I already explained that the plus sign of the delta G0 change in the free energy is represented by the plus sign. It indicates that it requires energy. And here the D0 value, delta G naught value, that is the standard free energy value of a system for one polypeptide bond is plus 21 kilojoule per mole. And the length of the peptide bond is 133 angstrom. You understand properly, and if you feel any problem, you are free to write me, and I will again explain, because this point is most important for the proper understanding of the Ramachandran plot. And here the bond is 133 angstrom. The first carboxyl group of first amino acid is associated in the formation of polypeptide bond. An amino group of the subsequent amino acid is responsible for the formation of the polypeptide bond. And due to the negativity of oxygen and positivity of nitrogen in the subsequent amino acid, the partial negative charge, that is represented by delta negative, is created on the carboxylic group, matlab O, associated with the first carboxyl group, and in the second subsequent amino group, the delta positive charge is existed. And if we considered Cn independently, if we considered Cn, that is the covalent bond present between carbon and nitrogen, it is necessary to understand here properly. The nature of the peptide bond is 1.33 angstrom. The partially negative charge is developed on the oxygen associated with the carboxyl group, and the partially positive charge is developed on nitrogen associated with the subsequent amino group of subsequent amino acid. And if we study the carbon, the covalent bond associated with the carbon and a nitrogen is a single bond, then it represents 1.49 angstrom. And if the carbon and nitrogen represents double bond, then it is of 1.27 angstrom. So the 1.33 angstrom of peptide bond is in between the single bond and double bond existed between carbon and nitrogen. So the peptide bond represents partial double bond character. And if we analyze, then the double bond nature of the peptide bond is 40%. Now, due to the double bond nature or the electronic arrangement of the carboxyl group, molecules associated with the carboxyl group and molecules associated with the amino group, oxygen has a partial negative charge and nitrogen has a partial positive charge. So, if we consider the molecules around the peptide bond, then we observe the 6 atoms. One is the alpha carbon of first amino acid. Second is the carbon of carboxyl group. Third is the oxygen of carboxyl group. Fourth is the nitrogen of amino group of the subsequent amino acid. Fourth, the hydrogen associated with the amino group of the alpha carbon of the subsequent amino acids and alpha carbon of the second amino acids. So, if you consider the one plane, it represents the 6 atoms around the peptide bond. And they are unipolar. They are unipolar. polar. As I think that you all properly understand my explanation, now we observe the torsion angle, or and just I am representing it by the pen. You consider like this. Suppose here it is the pen and it is represented the peptide bond. One alpha carbon is here and one alpha carbon is here. And now the alpha carbon, sorry, nitrogen is here and carbon is here. That is 6 molecules. Nitrogen is here and carbon is here. Now the OH and N is associated. Suppose here the OH of the alpha carboxylic acid and here is the H. Here is the OH and here is the H. In this way, it represents the three-dimensional structure. And the angle formed by the oxygen and nitrogen represents the torsion angle because it has the flexibility. Just I am going to explain. I am representing it by a pen to you and for your proper understanding. Suppose here the carbon of the first alpha carbon, that is the carboxyl group, carbon of the carboxyl group. Here the nitrogen of the carboxyl group. Here the peptide bond, it represents the peptide bond. One oxygen is associated here of the alpha carbon and the hydrogen of the subsequent amino acid is represented or associated by like this. So the angle formed by the oxygen with respect to the peptide bond and the angle formed by the hydrogen with respect of the peptide bond represents the angle of torsion or torsion angle. And here the torsion angle, that means the partially developed charges and the torsion angles are defined by 4 points. Just I am represented 2 points between the peptide bonds, 1 point of the carboxyl group and 1 point of the hydrogen group. In this way, it represents the spatial arrangement. So, the rotation of peptide bond in this condition is restricted. Here the peptide bond does not represent the movement, or it is non-flexible because the rotation of peptide bond is restricted. Why restricted? Due to the double bond nature. Of course, it appears as a single bond, but 40% peptide bond represents 40% nature of double bond. So, the double bond movement of the double bond is restricted. Then, if you observe the two planar structure with respect to the oxygen and nitrogen, just I am representing by representing by the use of pens, it represents the cis and trans nature. If both are on the same plane, that the torsional molecules are on the same plane, it represents the cis, and in the reverse, if it are opposite, it represents the trans. So, the restricted rotation about C and N bond, that is peptide bond, can be specified by omega. It is represented by omega. And just I am representing like this and this one. So, in the cis, omega represents 0 degree angle. And in trans, it represents 180 degree. So, the value of omega is 0 for the cis configuration and 180 for the trans configuration. Of course, the trans configuration is favored in the natural system. However, proline, because it is present in the amino form, not in the amino form. However, proline resides or protein residues prefer cis configuration. That means, except the proline, due to the amino group, the nature of the bond is different. So, in the except the cis form, the majority of configuration is present in trans form. So, the angle, that is the omega, that is represented the omega, and the two torsional angles, here the two torsional angles is present. Just I am representing by the use of pen. One torsional angle is associated with the amino group, and another torsional angle is associated with the carboxylic group. And the torsional angle associated with the amino group is referred as phi, and the torsional angle associated with carboxylic group is referred as psi. So, the graph is represented phi, psi and omega graph, or Ramachandran graph, or Ramachandran plot. So, if you consider the theoretical aspect, if we consider the theoretical aspect, the phi and psi have any value between plus 1 degree centigrade, plus 180 degree to minus 180 degree. Any value. Theoretically, we have chance to obtain the torsional angle of the amino group or torsional angle of the carboxylic group, or in another way, symbolically we represent the phi and psi angle. We have chance of any angle from plus 180 degree to minus 180 degree. However, not all combinations are possible. Why? Because due to the physical classes of atoms in three-dimensional space, each atom has a specific configurational possibility in a space due to the physical classes. So, theoretically we have chance, but the angle is determined by the molecules associated with the torsional angle and their behavior, the molecular behavior in the space. Two atoms, here the possibility that two atoms cannot occupy the same space at the same time. If any molecules, if any space is acquired by any one atom, then there is no possibility to second atom occupies the same space. So, two atoms cannot occupy the same space at the same time. These physical classes are called steric interference because the molecules have the specific class and the specific molecules acquire the specific space in the configuration during the spatial organization So, the one class interfere to the spatial arrangement of the another class sometimes, and that interference is referred as steric interference. And most values of phi and psi are therefore not allowed to steric interference between non-bonded atoms. If the atoms, because we observed here the association of bond between carbon, oxygen, nitrogen, and hydrogen. But in each amino acid, some other atoms are present having no role in the bond formation. But without taking part in the bond formation, they require some space. So here I am saying that most values of the psi and phi are therefore not allowed due to steric interference between non-bonded atoms. The atoms which are not involved in the bond formation, but they require a space. And that space, the requirement of a space of the non-bonded atoms determine the spatial arrangement also. And they provide the steric interference. These permitted values can be visualized on a two-dimensional plot. That interference we can visualize in the two-dimensional plot. If you are plotting the molecular arrangement in the two-dimensional nature, and that two-dimensional nature represents the spatial arrangement and distance or specific nature where the molecules are present in the form of the bond angles and bond lengths or association lengths. These plots is called Ramachandran plot. And that means if we are plotting the Ramachandran plot, or if you are representing the Ramachandran plot, that plot is associated with phi and psi on x and y axis respectively. And in this way, we study the Ramachandran plot. So, here, if you observe the Ramachandran plot, we studied that the amino acids are present in D form and L form. And then a specific isomeric nature of the amino acids, that is the D form and L form, have different orientation. And due to the different orientation of the side chains of D and L amino acids, they represent different nature of the Ramachandran plot. So, the Ramachandran plot represents the molecular arrangements, and they are represented in the two planar structure. And the two planar structure or two planar plotting is representing the spatial arrangement of a specific proteinous configuration in the form of angles phi and psi. And as we discussed in my last class, that Proteins are present in the primary structure, secondary structure, tertiary structure, and quaternary structure. They all configurational proteins represent variation in the phi and psi, that is in the torsional angle. As we discussed in the primary amino acids, I am going to represent some angles of the primary and secondary proteins. I have already discussed the primary protein. And here, if we studied in last class that the collagen is present in alpha helix structure, and in the collagen the phi angle is minus 51 and psi angle is plus 153. It represents the torsional orientation, not the peptide, because I am saying that the bond present between the carbon of the carboxyl group and nitrogen of the amino group is non-torsional, non-flexible. So the torsional angle in the case of collagen is alpha, phi and psi. The phi angle is minus 51 and psi angle is plus 53 in the case of collagen. And when we come at the helical structure, and if the right-handed alpha helix is present in the secondary structure, the phi angle is minus 57 and psi angle is plus 47. And if the alpha helix is right-handed, because in last class I am saying that the alpha helix may be left-handed and may be right-handed. Of course, in the nature, the left-handed form is more abundant. But if the alpha helix present in the right-handed, the angle nature is the same. Angle value is same, not nature. Angle value is same. That is for phi, it is 57 as the alpha helix. The psi is 47 as the alpha helix, but the rotational direction is changed. And in alpha helix, the phi is minus 57, but in left-handed beta helix, the phi value is plus 57. Whereas the psi value is plus 47 in both left and right-handed alpha helix. The second secondary structure we observed was the beta pleated structure. And in the beta pleated structure, the phi value is minus 139 and psi value is plus 135. But if the beta sheet is parallel, because in last class I told you that beta structure may be anti-parallel or may be parallel. So in the anti-parallel, the phi value is minus 139, psi value is plus 135. And in parallel pleated, beta pleated structure, the phi value is minus 119, that is 119, and psi value is plus 113, that is 113. So here we observe that the bond is same, arrangement is same, parallel is same. In spite of all the similarity, they represent a variation in the spatial arrangement. And for example, I am describing the variation in the phi and psi value in some secondary configurational proteins. So, the Ramachandran plot is most important topic for the analysis of proteinous configuration in space, and it has it has large number of significances for the analysis of the functional aspects in the biological system. Of course, they are also used for the therapeutic values. So, the Ramachandran plot is necessary. It is necessary to understand properly the nature and fundamentals of the Ramachandran plot. I think that you all understand that what is the Ramachandran plot, what is the bonding nature, why the bonds associated, why peptide bond is not flexible, why the torsional bond, that is the phi and psi, are flexible, and how we can explain the spatial arrangements of the atoms in the molecule in protein. Okay, if you have any problem, any viewers or any students has any problem, you kindly give me message. I am just waiting for 1 minute. If you have no problem, then kindly give me message that no sir, I have no problem, or I have no problem, because I just want to understand that you are properly understanding the topic, or you have any problem. So majority reply that there is no problem. Someone write me that sir, I have question related 7th May class. Yes, you can ask. I don't know because name is not mentioned. If you have any person have a question or have any problem for the 7th May class, you are free to give me message on just on my YouTube of the 7th May class. I will reply you certainly.